Nature has reminded us recently that we humans are weak in many aspects. Parts of nature can take advantage of weaknesses in the human constitution and cause us to fall foul of terrible diseases, whether they are infectious diseases or chronic diseases of life and aging. Can we humans do better? Of course we can. It's in human nature to try and do better than what we have inherited from our surrounding. Humanity has been the story of engineering. Humanity has been the story of medical research. Humanity has been the story of education. We have transformed a lot of the situation we have received from our forefathers and foremothers. But where are we going next? What we're discussing today is what we can do with the relatively new field of genetic editing. And I'm joined by three panelists who I'm now going to ask to briefly introduce themselves. Jamie.
Thank you so much, David. Thanks, everybody. Really thrilled to be here with you. I'm Jamie Metzl. I'm a technology futurist and a senior fellow of the Atlantic Council. I'm also a member of the World Health Organization International Advisory Committee on the Future of Human Genome Editing. I'm the author of 5 books, including most recently Hacking Darwin, Genetic Engineering, and the Future of Humanity. And I'm the founder of One Shared World, which I will tell you all about in just a few minutes.
And your previous books, Jamie, were they all science books?
No, mixed. Before Hacking Darwin, my most recent 2, Genesis Code and Eternal Sonata, were near-term sci-fi novels exploring all of the issues that we're going to talk about today. Before that, my first 2 books, one was a history of the Cambodian genocide, and one was a historical novel dealing with all of the very complicated issues surrounding genocide and human nature and the worst devils of our nature.
Wow, I was in the Cambodian refugee camps a couple of years ago. We can compare notes some stage. Nessa, would you like to introduce yourself, please?
Sure, great to be here. Lovely to see everyone. My name's Nessa Carey. I'm a visiting professor at Imperial College and also an entrepreneur in residence at the University of Oxford. I worked for 15 years in the drug discovery industry, and I write popular science books. So my first was the Epigenetics revolution, my second was junk DNA, and my third was Hacking the Code of Life. So always tending to try and communicate at the cutting edge of the funkier bits of genetics, and really looking forward to today's discussion.
Are you working on a 4th book? Can you say anything about that?
I have 2 ideas in my head at the moment, which are at completely opposite ends of the spectrum for each other, but I'm frankly still just recovering from all the public engagement work on Hacking the Code of Life, which has been pretty overwhelming.
Well, hopefully it won't be too overwhelming today. Thank you.
Thank you, David, and thank you, Nessa, and thanks, Joyce, and thanks to all of you for being here. This is a perfect start, actually, to what we're going to be talking about today, because David mentioned that we're all weak, and that's a problem, but our species is unbelievably strong, and now we have these powers that we have for millennia attributed to the gods. And there's 2 problems. One, is our technology doesn't always work perfectly, as we've just seen today. And 2, in having these powers, we need to make sure that we have the wisdom to use them wisely. So what I always say is, at their core, these aren't conversations about technology. Technology brings us to the conversation. These are conversations, and must be conversations, about what are the values that we will use to guide the application of these incredibly powerful technologies. And we come together at a really critical time for our species. And everybody obviously is talking about and thinking about the pandemic. But this pandemic isn't reaching us in a vacuum. It's reaching us in the context of this historical moment, and something much, much bigger is happening. And that's why I think that it's important for us to take a step back and look at 2 converging trends around this moment. The first, which is the topic of our conversation today, of our technology. I mean, it's crazy for us to believe that although Gregor Mendel had his first discoveries about genetics in the 1870s. It was really around 1900 that knowledge became better known around the world. And in this last 120 years, which is the blink of an eye in historical terms, we've come just incredibly far. And we've gone from just a basic understanding of genetics to now having this ability to read, write, and hack the code of life. And that it's an awesome power. And it comes with just an incredible and critically important responsibility. And if it feels like the speed of technological innovation is speeding up, fasten your seatbelts, because it's only getting faster. 100 years ago, we had about 2 billion people on Earth with a 20% literacy rate. That means about 400 million people able to contribute to the field of widely shareable knowledge. Now we're 7.7 billion, 85% literacy rate. That's about 6.5 billion people who are able to be part of this process, contributing brain power. We have a super convergence of technologies, so that with every innovation, we're speeding up other innovations in even what seem like unrelated fields. And everything that we're talking about is connected to the computer revolution, the AI revolution. All of these revolutions are coming together and speeding up this process. And so on one hand, we have this genetics revolution. We've gone from the $1 billion sequenced genome in 2003 to around $600 or $700 for the whole genome sequencing now, and it's trending towards negligibility. We're seeing this transition of our healthcare systems from generalized to precision to predictive medicine, healthcare, health, and life, with all of those implications. We're seeing a transfer of thinking about genetics as something that happens through the medical system to something that happens broadly across the population. It starts with DIY genomics, 23andMe, but that's going to grow a lot, and we're going to have all kinds of information that's not going to be mediated through our healthcare system, but it's going to tell us a lot of information that we will at least interpret as saying something about who we are, about who our children are, about how we may parent. And then, as Joyce knows, and we are going to see and already seeing a fundamental transformation of not just the way we make babies, but the nature of the babies that we make. And we'll see, and just from, we did here in New York, actually in this table where I am now, we did a 40th birthday party last year for Louise Brown. And we had the whole, the IVF menu with tapioca and deviled eggs and other appropriate food items. But it's crazy that was 40 years ago. And I certainly, when I write about the end of procreative sex, that doesn't mean we're not going to have sex, but more and more, we're going to have kids through IVF and embryo screening. And ultimately, and I think not that far into the future, very, very limited, but increasing amounts of genome editing of pre-implanted embryos, initially to remove very dangerous single gene mutations, but it's not going to stop there. And there's no natural dividing point between therapy and enhancement. And so that will grow over time. And all of this will be fueled by just our differences, our diversity within and between societies, by competitive pressures, by societal norms of people imagining a certain kind of child as what they would like. And maybe they're getting very strong societal cues about what that is and isn't. And we need to be very, very mindful that these aren't some kind of objective truths. These are all, we live in societies with norms. So my book, Hacking Darwin, is about this, and about how if this science is advancing extremely rapidly, but we need to be having the kinds of informed dialogues about how best to use it. In the UK, I tell everyone, I think you're the best for these types of issues. UK, I think, is the best regulated country in the world. I would love to see the HFEA model exported, or at least shared, and every country should have some kind of version of the very responsible regulatory system that you have there. It's not coincidental that the UK Biobank has become the most impactful biobank in the world, even though it's not the biggest, and it's because of the high standards you have in the UK. So that's one trend, is this technology. But there's another trend that's converging on this moment, and that is a broader trend of history, and which connects why we're in this terrible, frightening moment around the pandemic. In 1918, 102 years ago, we were in a very similar moment to now. During the war, 5 major empires had collapsed, and there were people who were coming together who were imagining what the world to be reborn would look like. And everybody recognized that the old world was broken and had broken, that there were these new technologies that were driving change forward, that were creating the opportunity to organize in new ways, to communicate in new ways. And this is just mass oversimplification. But the progressives around Woodrow Wilson and 14 Points, that whole movement failed because the case for inclusion and for construction, they lost the battle. The people who were advocating for narrower identities, the nationalists, the fascists, they won. They organized themselves better, they started marching quicker, and they won that game. And that's what brought us the Second World War. At the end of the Second World War, the forces of inclusion, of construction, came together, backed by the might of the United States and others, and they built a superstructure that was designed to temper the worst impulses of rigid sovereignty, World War I, and excessive nationalism, World War II. And that's the world that we've lived in until now. And now 2020, I see this historically as the end of the post-war period. I think that's what we'll see. The 1648 to 1945 was the period of the nation state at its height. Then 1945 until 2020 was the period of the nation state, which tried to be tempered by international institutions like the United Nations, which ultimately failed because the states weren't willing to give up the sovereignty that was necessary to let the United Nations do the job that it had been asked to do. And certainly here in the United States, there's 3 big reasons why so many of us are dying. The first is the monumental Chinese screw up and cover up, particularly in the first 3 weeks in Wuhan. The second is that we collectively hadn't created a World Health Organization with the resources, power, mandate, and mission to actually solve the problem, which it easily could have if it had a global surveillance system and immediate deployment of emergency response systems. And then here in the United States, where we've had basically a fundamental breakdown of governance and civic culture, and we're dying. Taiwan had the same information as we have here in the United States. They have 6 COVID deaths. We're already past 100,000. And so with that moment, there needs to be a fix. I mean, we need to recognize the power of our technology, and our technology, I think, will give us some answers to this smaller problem of COVID-19. I mean, we'll get better treatments. We will get eventually a vaccine, again, because we have so much brain power and because biology is readable, writable, and hackable. And so we will find this hack. But we're not going to solve the problem. We may solve the problem of COVID-19. We're not going to solve the problem of pandemics. We're not, frankly, going to solve any of these big global collective action problems unless we do the right analysis and then come to the right solution. And so the right analysis is there's a mismatch, in my view, between global problems and national solutions. And unless we have a global collective human response to global problems, we'll still have the pandemics people trying their hardest and can't solve pandemics, and the climate change people can't solve climate change, and the WMD people can't solve WMD. And so that's why I mentioned One Shared World. So I have founded, and we have now thousands of people in almost 100 countries working together on this initiative movement called One Shared World. The URL is oneshared.world. And what we're trying to do is to create an inclusive global social and political movement representing the common aspirations of humanity, because unless we fix the superstructure problem, it's going to be very, very difficult to solve any of the sub-problems. And so we've drafted a declaration of interdependence that you'll see on the website. It's translated into multiple languages. We have lots of people volunteering for more. Diane has a great question: why not reform the UN? It's a really essential question. We need to support the UN, but unless there is a popular movement demanding that our nations create a UN that can do its job, our countries aren't going to do it, because our political leaders have done the exact job we hired them to do, which is protect our national prerogatives. We have to change that, and that will change the UN. Just supporting the UN alone will get the UN we have, and that's insufficient. But it's a great question, Diane. Thank you. And so we have to use the technologies that we have, and we will, to develop a vaccine. But we're talking about hacking, and there are different systems that we have to hack. We have to hack this virus. We have to hack our healthcare system. We have to hack our world, because the world was already broken before this virus happened. It was totally comfortable for everybody that we had 3 billion people living in abject poverty. And now we're recognizing our interdependence. We realize, wait a second, that's a vulnerability for me. Those 3 billion people are 3 billion viral vectors. And so if things get better here in the United States over the summer, and the virus migrates to the southern hemisphere and mutates and grows and then comes back here in the fall, I'm dead. And so my health and well-being reside in not just every other person, but in the ecosystem that we share. And I think that for this community, what does it mean to be a futurist? It's not just saying, well, here's what the future may look like. It's, here are some different scenarios of what the future could look like. And once we lay out those scenarios, very quickly, we realize our responsibilities to fight for the best possible outcome. So let me just end there. Thank you.
Wow, so you have looked at the technology, and then you have looked at the history and the geopolitics. And I think we will need to look at both of these questions as we go through this discussion. I'll refer everybody to the poll that was set up at the beginning. A few of you have answered the poll. If you click on the poll button near the bottom of the screen, you will see that actually the biggest set of answers to the question there about which of the following statements about genetic engineering of human babies do you most agree with, it is that there will be meaningful international agreements on genetic engineering of humans, and these agreements are both desirable and achievable. That the issues that genetic engineering of human babies brings up are so large that they can't be addressed by individual countries, but at least the audience, the majority, are saying they are optimistic that this can be done. Well, let's put that poll away. Nessa, I'm going to give you the floor. And by what topics would you like to feed into the discussion at this stage?
Yeah, so I want to build on some of Jamie's comments and expand them as well. When we talk about hacking the code of life, there's a real tendency to everyone to jump immediately to how we do that in humans and what it means for humans. It's incredibly important to realize that this technology can be used in loads of other ways, many of which will have much greater impact. So it can be used, for example, to improve food crops, to improve their resilience, to improve yield. Now, if we can do those things and do them well, we could end up in a position where we can feed the world while increasing biodiversity, i.e. by using a smaller amount of the land for agricultural use, giving the natural world more space. If we do that, we immediately start to decrease our risk of future zoonotic pandemics. We immediately start to create a situation where there's less chance of viruses skipping from other species into humans. And actually, the implications of using gene editing in other species are, I would say, in some ways, much more deserving of great ethical debate than about humans. And one particular thing that we really should be focusing on is the use of gene editing with gene drive to try and attack certain invertebrate species, because we could have ecosystem effects there. And that technology combination needs to be really carefully controlled. If we talk about humans, though, and I think we do that because we're very, very individualistic. we need to think about two different approaches. And these are the fact that you can edit what we call somatically, which is the body cells of somebody, to potentially treat them for a disease. Essentially, what we're doing is we're just using genetic engineering, gene editing, to alter someone's DNA so they no longer suffer from a devastating disease. It's actually just another way of doing drug discovery and drug delivery in many ways. It's a new technology, but we're essentially using it for the same purposes. Now, we have extraordinary initial results, by we, I just mean the world, in treatment of things like sickle cell disease and thalassemia, hemoglobinopathies. This is already being used in humans. It's in clinical trials, and this is less than a decade after technology was actually identified, which is extraordinary. The results are so startlingly good that it looks like for some diseases like sickle cell disease, this won't just be a treatment, this will be a cure, and that's extraordinary. We're also seeing clinical trials using gene editing to create particular type of immune cells that will fight cancer. And again, the initial signals are looking good. It's certainly safe to use. That sounds fine. But one of the things that might be very different about gene editing from standard drug discovery is in standard drug discovery, if a patient reacts badly or doesn't like the effects of a drug, you can stop giving them the drug. If you've edited their DNA, it's much harder to reverse that change. Now, it's very difficult to imagine somebody with sickle cell disease saying, do you know what, I'd like to have sickle cell disease symptoms again. That's very unlikely to happen. But if we were to start thinking of using this technology to perhaps tackle certain forms of intellectual disability or neurodiversity, there might well be consequences to a treatment that we couldn't reverse. If you look at the history of certain fields, such as drug treatments for Tourette syndrome, and some other conditions similar to that, sometimes patients will take themselves off the drugs because they find the side effect outweighs what they perceive as the benefit. And that might be quite difficult if we were to do that in genetic treatment of humans. But the important thing about that kind of treatment is that it's only done to people who will have that treatment, the gene editing, and they will not pass it on to their children, because it will only have been done in their body cells, say the bone marrow cells of people with sickle cell disease. The other much more contentious use of gene editing is in what's called somatic gene therapy, which Jamie mentioned. Germline gene editing. And in germline gene editing, you would be treating a very early embryo to remove a mutation. And all the cells of that offspring, of that embryo rather, as they grow and develop, they'd all contain the correction. And also that person would then pass on that change to their offspring if they have kids. Now, we have never deliberately done that before. We've never had the technology to do it. It's perfectly doable with existing technology. It has actually been done in China, very poorly regulated, total disaster in terms of the ethics and the movement of the field forward. But there is nothing really technically that is likely to stop this in the future. Now, some people really, really worry about this. They say it's playing God, that it's changing the DNA and we don't have the right to do that. And I think that's a very interesting position. I think one of the things that the capabilities of gene editing has thrown up is that we are all incredibly possessive about our DNA sequence, even though most of us don't know what our DNA sequence is. It's almost like, it's my genome, it's who I am. Well, it's not who you are. Who you are is dependent on a huge number of features, not just your genetics. One of the most telling statements I've heard is from somebody who has a devastating disease in their family. And they have basically said, why are you having such a fuss about germline gene editing? Because all you would be doing would be changing one tiny sequence of DNA in my child so that that sequence was the same as the other 8 billion people on this planet. And when you put it into that context, that's actually quite telling. And one of the things that we have to address with gene editing is it has completely flipped the technological questions that we can address because it's so good, but it's also flipped the ethical question. The question that we always used to have with almost any technology for medical health was, do we have the right to do this? Especially if we were talking about something very dramatic. The technology is so good and getting better all the time that I think in many forms of gene editing for humans, we now have to flip the question and say, do we have the right to withhold it? What gives us the right to say to families or to individuals with devastating disease, you know, we could cure this, we could fix this, but we're not going to. Oh, have you just lost me?
You're still here.
David, am I still
We
there?
can hear you. We can hear you.
Okay. Okay, I'll carry on talking then. I haven't got much more to go.
I don't know why your video's gone, but let's continue.
No, me neither. It could be Norfolk has the worst broadband in the world. I mean, it might just be the 2 tin cans and the piece of string linking me to the exchange have fallen over. So we do have to question, do we really have the right to withhold this treatment? However, what we also say is if we have the right to withhold it, or we have the right to give it, who do we give this treatment to? We will need to address social inequalities in healthcare. And that is a very big issue, but it's not specific to gene editing. That's also true of any other expensive drug. The other area that people worry about, of course, is designer babies. This whole issue of where do we go from cure or treatment of a disease to designer babies? I don't actually worry too much about the designer aspect of things because most of the things for which we know people would try to select are stuff such as height, beauty, which is very difficult to put a number on, intelligence, sportiness. All of those things are influenced by a huge number of genes acting in complex ways with the environment. Gene editing is fantastic and it's a brilliant technology, but it can't go in and change hundreds of things all at once. So I don't worry about that sort of designer baby issue. Where we will have to address this is when we're not at the extremes of pathology, when you're no longer looking at life-limiting or life-ending conditions. If a condition is going to be lethal, then anything you do with gene editing is unlikely to make the situation worse. And everyone would agree that it's, say, a lethal, devastating disorder. I could give you hundreds of examples. But what do we do about something like deafness? Some people might say that deafness is a devastating disability. Many members of the deaf community will say it's actually not a devastating disability. It is a complete pain to deal with simply because society does not make appropriate adjustments. And also, profoundly deaf people will typically use a sign language, and sign languages are diverse throughout the world. They are linguistic groups. If we start using any technology to get rid of those linguistic groups, we start running into possible definitions of genocide. So it's in those fuzzier areas where there is debate about whether we have a social or a medical model of disability that we need to be very careful. The other area where I think it's going to be vital is the issue of health tourism. We can see that there will be labs offering dodgy gene editing cures and treatments for devastating conditions, taking advantage of people's desperation, even though they can't really improve things. We already see this with stem cells. We see stem cell tourism, and we will see this, I am pretty sure, with gene editing. And it's rumored there are even in vitro fertilization labs around the world in occasional countries who are offering that they can help you get the kind of baby that you want. They won't be able to, but they will take advantage. And the very fact that they're saying they can do this lends credibility to the concept that it's an okay thing to do. So I think that is very, very dangerous. So lots there, and I'm going to stop because otherwise I'll just keep going forever. So I'll stop now.
Well, we'll bring you back into the conversation because there's so many interesting points you've raised as well, Nessa. Hopefully, maybe you could unplug and plug back in your video camera, whatever it needs to do. Fascinating discussion about the deaf communities, health tourism, and so on, and about gene editing in other species for the sake of better agriculture. We'd love to dig into some of that shortly. But let's turn, for the time being, to the third panelist, Joyce Harper, Professor Joyce Harper, and I'll give you the screen to make your opening remarks. Joyce.
Right, I've just unmuted myself. Yeah, somehow I'd got cloned. Between the green room and opening of the conference, I'd actually got cloned and there were two of me. So that's why we were getting all that feedback. So sorry about that. So just my short introduction first. I'm Professor of Reproductive Science at the Institute for Women's Health at University College London. and I did a PhD at King's College in London and started a career as a clinical embryologist back in 1987. And in 1992, I decided to get back to research, and I joined the team that were performing the world's very first pre-implantation genetic diagnosis cases. I worked with Alan Handyside, Robert Winston, and Eleanor Contongiani. And Alan and I published the world's first data on PGD paper in 1994, and I wrote another paper on the world data of PGD in 1996, and then in 97 became co-founder of the International PGD Consortium. And I published the first and second ever textbook on PGT. I'm afraid they're very out of date now. We were going to revisit them a couple of years ago, but I never quite got round to it. But now most of my work is dealing with fertility education. I'm co-founder of the UK Fertility Education Initiative, and last year I set up a global fertility education consortium. So that's my short introduction, David. Did you have any questions? Great. Shall I just continue?
I think please do. I'm sure you've got views on what you've heard the other two say.
Yeah. So it's been really interesting. I'm really looking forward to today's discussion. So I've worked in this field for over 30 years, and throughout this time I've become very concerned about the ethics of reproductive genetics and where this would take us. And one of the main talks I give at international conferences is about the future of reproduction and reproductive genetics. And I want to give you a summary of that talk in the next 5 minutes. And I end the talk by asking the audience, how will our future children be made? Will women freeze all their eggs? Will all women freeze their eggs? Will we use PGT to select the most desired embryos for everybody? Will we use genome editing to make the perfect child? And will we use an artificial womb to carry the pregnancy? So as I said, I'll just quickly go through how I give this talk and what I explain. So I'm going to give you some science, just briefly. What is this procedure, pre-implantation genetic testing, which we used to call pre-implantation genetic diagnosis? So PGT actually started in 1989. That was when the first cases were done at the Hammersmith Hospital. And as I said, I joined the team in 1992. And at that time, we were working specifically on trying to prevent genetic disease for couples who are at high risk. So the technology was very basic then, and we used simple techniques to sex embryos for couples carrying sex-linked diseases. So we replaced the female embryos, which could be carriers or free from the disease, and we didn't transfer the males. And around that time, when I joined, we also started working on PGT for cystic fibrosis. So we've really moved on in the last 20, 30 years in this technology. Certainly, it's going exponentially. So I used to be involved with the embryo biopsy procedure, and one of the very basic diagnostic techniques back in the 90s was a technique called fluorescent in situ hybridization. And we were one of the first to publish the problem we see with human embryos. They can be quite mosaic, and they can also have chaotic chromosome arrangements, and this has caused some problems with the diagnosis over the years. So one of the very controversial issues in pre-implantation genetic testing has been a procedure that was called pre-implantation genetic screening. It's now called PGT-A for aneuploidy. And this technology, PGT-A, uses the same techniques we use in PGT, but it's applied to infertile couples going through IVF. So it's not being developed specifically to look at genetic disease or couples that are at risk of transmitting genetic diseases to their children. It's to try and look at the chromosomes in patients going through IVF to improve the success of IVF. But unfortunately, over the last 20 or so years that this procedure has been offered clinically, in my view, we haven't had any conclusive evidence to show that this procedure does actually improve live birth rates. And I'm using this as an example of some of the issues I think we have to think about these new technologies. So I am part of the Human Fertilization and Embryology Committee that developed a traffic light system for patients to understand how new technology that claims to improve success is actually effective. So this HFEA in the UK, I know some of you are not in the UK, this is the body that governs IVF treatment in the UK. So the HFEA committee awarded PGT-A a red traffic light, which means that at the moment, the committee did not feel there was conclusive evidence that this would be something that would improve IVF success. And this year we published a paper comparing UK and USA PGT data. And we've now seen that there are a growing number of IVF clinics in the USA that are using PGT for every single IVF patient that comes through their door, that goes through for treatment. So this has brought in the huge discussion about the commercial aspect of this technology. So there is no doubt that IVF genetic testing has a huge financial gain for many companies and a huge financial cost to the patients. So if we look at some of the controversial uses of PGT besides PGT-A, it's illegal in the EU, but sexing embryos for those patients who want to choose the sex of their child is offered in other countries, including the USA. So these patients don't need to go through IVF to get pregnant, but they do use it because they want to choose the sex of their child. PGT for polygenic disorders is already here. I was hoping some of our colleagues from Genomic Prediction would be online, but I haven't noticed them in the group. There may be some whose names I don't know. There are also companies that are offering PGT for eye color. And when I talk about this, people say this is not possible, but it is possible. And there is no doubt that choosing an embryo that is the healthiest or has some desired characteristics is really imminent. And as long as couples have the funds to do it, they can do it. So I say this hesitantly, but Gattaca really is here. So fertility experts have been thinking about these ethical issues around PGT for many decades, and I've led 2 meetings of experts from the European Society of Human Genetics and the European Society of Human Reproduction and Embryology, and we look regularly at the interface between genetics and reproduction, and we've published these papers by open access, so you can find these online. And in our last meeting of the group of experts, we talked extensively about genome editing of human embryos. And I was very lucky enough to be a member of the Nuffield Council on Bioethics working group on genome editing. And we published our report last year. And our conclusions were that genome editing could be used for therapeutic genetic interventions, such as couples carrying a genetic disease. But we have to remember that they do have other options. They can opt for PGT, or they can opt for prenatal diagnosis. And the Nuffield Council committee, we could not dismiss that genome editing could also be used for genetic enhancements. And I think that's something we will talk about now, such as eye color, perfect pitch, etc. There are genes for these. So this would be when couples don't carry that characteristic, but they want a child with that characteristic. So we made 3 recommendations. We felt we needed public debate to see if the public really want this procedure. And if they do, we need to have the science to ensure the procedure is safe, and legislation to set the rules on if and how we use this technology. So this is going to be the first time that we are ever going to be able to manipulate the genomes of our future children. And I think we have to think very carefully about what are we doing, why, how we're doing it, and why. And if we edit our kids to be highly intelligent, sporty, or musical, and they decide they do not want to live up to their genetic editing, are we putting them under pressure to conform to our ideal? I also last year took part in an Oxford Union debate. This house believes that genetic engineering will undermine the nature of humanity. I spoke for the motion and I lost spectacularly, and I was really not surprised. I agree with my fellow panelists that in the near future, the use of IVF and genetic technology will increase, and we will see less people conceiving naturally. In his book, Jamie describes himself as a technology optimist. I have hesitancy about our reproductive future. I think we need more discussions such as we're having today, and we need more efficient ways to ensure that this technology is safe for future generations. Jamie in his book said that the UK proved mitochondria transfer was safe, and I do hate that word proven, and I think we need more work to prove anything. So I want us all to stop and think. Just to finish off, female reproduction is taking an interesting turn. Today my main work is on fertility education, and women are having children at later ages in every country. We still have female fertility, which is declining with age, significantly so from age 35. And yesterday I launched a survey to ask women about their attitudes to having children. So what should our future be? Should all women have egg freezing? As our female students graduate, do we send them off to the IVF clinics to freeze their eggs? Should all the companies offer their female staff egg freezing? And what about the growing number of celebrities using a surrogate to carry their children? If we can afford it, should we use this technology? I have 3 children, all born by IVF. I know it's very difficult to go through, and it worries me that this will become the norm. This technology is expensive. An IVF cycle in the UK is about £8,000. It's even much more in the USA. PGT is about another 3,000 in the UK, and to freeze 3 cycles worth of eggs in the UK costs about £15,000. There are many couples in the UK who are infertile but cannot afford IVF treatment, as the NHS does not even fund 1 cycle in some areas. Maybe we do not need IVF in the future, as there'll become a time when we can use stem cells to make eggs and sperm. Then PGT would select the most desired embryo, genome editing it for medical and non-medical reasons, and we'd have artificial wombs. I tell my students to read Brave New World, The Handmaid's Tale, and watch Gattaca. They need to think about how their children will be made. Will it be utopia or dystopia? Thank you.
Well, that's raised all kinds of questions as well. Thanks to all 3 of you for stretching our minds. I'm going to go around the 3 of you again, give any comments you've heard on what you've from each other, maybe slightly quicker this time, maybe up to 5 minutes each. Before I do that, I'll draw everybody's attention in the audience to the possibility you can ask questions too, and we will take the questions that have got the most upvotes in general. And if you want to ask a question that has a chance of being upvoted, I recommend you write short questions rather than very long essays. Even though your essay may be fascinating and technically correct, people's eyes probably won't take it all in, especially as most of the attention is going to be on the wonderful words shared by the panelists. So after one more round of comments from the panelists, I will start taking questions from the Ask a Question box. Jamie, I'm sure there's lots that you could say, but maybe if you can restrict yourself to about 5 minutes, please.
Sure. So first, thank you for those great presentations. I love them. I just want to agree wholeheartedly with something that both mentioned, particularly Joyce, and that is our need to have an inclusive public conversation and even decision-making process around these technologies. There's a tendency in the general public to feel, well, these are very technical issues. Let's just have experts make these decisions, and that's the wrong answer. I speak to the top scientists in the world at places like Lawrence Livermore National Laboratory here in the United States, and I speak to 7th and 8th graders in schools. My friend is the chairman of the board of a Hebrew Academy in Bergen County, New Jersey, and he asked me to speak to these 7th and 8th grade Talmudic scholars. And these 7th and 8th grade kids and the scientists at Lawrence Livermore all got the core questions that we are asking. This is not about technology. It's about what does it mean to be a human being? And what are our obligations to ourselves, to each other, and to future generations? And so if we want to move forward responsibly, it has to be done in a thoughtful, engaged, empowered way. And that's why I praised the process in the UK. Certainly, I didn't mean to say that it's totally proven, but the process that you went through before the Parliament voted to authorize clinical trials, it was very engaged, very informed, very inclusive, and it laid a foundation for moving forward together responsibly. There are still differences of opinion about what's the right step. That's why, as you know, in my book, what I'm calling for is a species-wide dialogue on the future of human genetic engineering, and it's not just a dialogue; it's a conversation that translates over time into norms, regulations, laws, treaties, and we're going to have to find common ground because, as I was saying before, we're all one species. We're all interconnected, and so all of these decisions, particularly when we're talking about germline changes, that touches all of us. So I really I agree with what Joyce was saying. The Nuffield Council has just done fantastic work, and you know the World Health Organization committee that I'm on. I've just been banging that over and over and over. We have to think broadly about governance, but we also need to think broadly about inclusion, and that means engagement, empowerment, dialogue, and a connection between dialogues and decisions. And then finally, just to coming back to Joyce's point from her debate, I think it's a very valid target to say this. I can understand how people would say what we're talking about here is changing some aspects of what it means to be a human. But I think we also all need to recognize that what it means to be a human is a moving target. We are biologically very similar to our ancestors just 20,000 years ago, but our lives would be totally incomprehensible to them. We are all superhumans with our vaccinations, with our ability right now to communicate with people half a world away, with our glasses that allow us to see. And so I think that it's not, and I say this in my book, the question is not, should we or shouldn't we play God? And I started with that, my remarks with that. We are in any conception that our ancestors have had about what gods do, we are gods. And so that's the great quote by Stuart Brand. We are as gods, so we better start getting good at it. And that's why it's ethics, but figuring out what the ethics are, what the guiding values and principles are, that has to be the work of everybody. And we have to change the way we think about governance to make that kind of empowered inclusion possible.
And briefly about polygenic traits, Jamie. And is it possible in the foreseeable future that we will be able to change enough genes simultaneously that we can have an impact on things such as IQ, if there is such a thing as IQ?
Yes,
or is that just science fiction?
I write about that in my book, and Joyce and I know well the people in genetic prediction who are talking about at least understanding polygenic traits. So the reason why I believe that embryo selection is the killer application, and especially if we can increase the number of eggs. So if we can have, basically, if we're using stem cell technologies, we can have an unlimited number of eggs. So let's just say that we have 10,000 eggs, which are derived using induced pluripotent stem cell technologies, let's say from a skin graft. So we have 10,000 eggs, fertilize them with the around 1 billion sperm cells that can be in the average male ejaculation, then have a fully automated process for growing them for about 5 days, extracting a couple of cells from each, again, fully automated in PGT, and then making selections from among those 10,000. In that case, you can definitely apply the polygenic knowledge of traits because the risk factor is relatively low. These are all, quote unquote, naturally occurring embryos. Once you start going into genome editing, and I have this debate with George Church all the time, but once you start going into genome editing and say, we're just going to go and we're going to hack a pre-implanted embryo and make 5,000 changes, I think that's going to be very, very difficult for a very, very long time, just because we'll probably, based on our knowledge of biology now and the foreseeable future, we'll just probably kill those cells. So I think embryo selection, in my mind, is the killer application. But then we will find, and George Church has an ongoing list of these, we'll find that there are a small number of single gene mutations, single variations that can have an outsized harmful or helpful impact. And I think that the number of those small changes will grow, and that will be, those will be the first targets. In 1,000 years, we'll be doing things very, very differently, and I'm sure it will involve very complex genome editing. But for now, that's how I see things playing out.
Many thanks. Nessa, what would you like to bring back to the discussion?
Yeah, so I'd certainly like to endorse that the Nuffield Council on Bioethics report on gene editing is absolutely exceptional. It's free to download. It's beautifully written. I would urge everybody to access it. It's a brilliant document. I think it's absolutely right that ethical discussions run ahead of where the science is at. It's a nightmare when ethics has to follow the science, and good ethical decisions are rarely made under time pressure. So I think it's really good that we debate these things, but I do think we have to be very careful that we don't put so much energy into debating things which will not be technically feasible for a very long time, if ever, that we then start muddying the waters for all kinds of genome editing questions. So I completely get that technology gets taken up much more than we expect. Louise Brown, 40 years ago, first IVF baby, everyone said that's going to be a niche application. There's now 4 or 5 million individuals on this planet who were born through IVF. So niche applications do tend to spread if there's a human desire for them. But I just find it very difficult to visualize a world such as Joyce describes, where every woman will be having her eggs frozen and everybody will be making huge reproductive choices through these very complex technologies. When we think about the fact that there's probably 500,000 women a year who can't get access to a toilet, I think the idea that they're going to be accessing this technology anytime soon is not really terribly realistic. So again, I don't think we should let the debate be dominated by those kinds of discussions. I think we also have to question, if we think about what Jamie was saying about embryo selection versus gene editing, why do we view them as something different? Why would we, for example, think it would be acceptable to choose or try to skew our chances of a particular phenotype through selecting embryos, but it would be somehow bad to introduce the same change through editing? What is this strange possessiveness that we have about our DNA sequence, that we don't want someone ever to change one of the 3 billion base pairs that we inherit from our parents? Why are we so obsessive about that? And I think that's an interesting question that we need to debate as a species. I think I agree entirely with Jamie about we've always played God. And as a consequence of that, we have become an absolute pest species on this planet. We are the most destructive species ever. And we are reaping the whirlwind of the seeds that we've sown ourselves. But we've always done it. So again, we go back to this issue of, if it's okay to prevent disability through a vaccine, for example, or through folic acid supplementation of foods for women who are planning to become pregnant, why would it be wrong to prevent disability through the use of gene editing? And I think perhaps the one thing that most of us haven't addressed yet in this debate, and is actually something that is absolutely vital, is that we need, and I know it's something very close to Joyce's heart, because again, I know the Nuffield report, is that we should be engaging the patient voice much more seriously. And patients of these devastating genetic diseases should be the people that we really, really listen to and engage, because they are the ones who know what it's like to live with these conditions. They are the people who know what it's like to have a son born with such severe neurological abnormality as a result of a genetic condition that the son has to be restrained his entire life so he doesn't self-mutilate while screaming in agony. That's just one genetic condition. So I think one of the things that we must do moving forwards is to be absolutely certain that the patient voice is at the center of any discussion about therapeutic cloning. This should not be something where scientists, clinicians, or any other group imposes these decisions either to make available or to withhold these treatments. So that's where I'll finish, is on a plea that absolutely patients have to be at the complete center and front of this.
David, if I could just jump in very, very quickly before, and I just wanted to clarify something that Nessa, so I certainly wasn't trying to make the case that there's something sacred or a huge difference between genome editing and embryo selection. The question I was asking was selecting for polygenic traits. And so I believe for polygenic traits, embryo selection is the first step toward that selection, just because we can't do it. We can't genetically engineer a pre-implanted embryo making 5,000 changes. We can hardly do it for one. And then I totally agree with what you said. We also shouldn't think that the human genome is so sacred that we wouldn't want to save some child who we know would otherwise die of a painful, deadly genetic disorder early in their life. I mean, my view, it would be immoral to have the ability to save that child's life and not do it. So sorry to jump in.
No worries. I'm hoping I didn't come across as suggesting that either of you were saying those things. It's I'm using it as the sort of, these are the issues that we have to have in these debates, because it is really complex and it does force us to think. I think it's actually forcing us to think much more about the use of reproductive technologies generally. It's just that gene editing is somehow, we're obsessing about gene editing in some ways, when perhaps there are either other applications of gene editing we ought to be thinking about more, or there's other aspects of reproductive technologies we should be thinking about more.
Joyce, what would you like to come back with?
Right, thanks very much. So it was a real privilege to be on the Nuffield Council Working Group on Genome Editing. It was a fabulous 2 years, and we did some fabulous meetings to discuss the various topics. And one thing we had not predicted was that anyone would be doing genome editing in a human embryo within the next few years. So this is one problem we have with any of this technology. It's as long as a piece of string of how long it will take. I can remember back in the early 90s, one of the main people working on gene therapy for cystic fibrosis, Bob Williamson, told me that gene therapy for cystic fibrosis was round the corner. This was about 1991. And we just can't predict. But what we do know is what is here now. So we do know that PGT is here. There are people using PGT already now for things like eye color, to choose the sex of their child. That's done, absolutely. And one problem we have now, as Jamie's talked about and talks about in his book, is that we are limited at the moment by the number of embryos we have. So we would be very lucky if we had more than 5 embryos to test by pre-implantation genetic testing. We're probably, five is a good number. We wouldn't be necessarily looking at more than that. So if you start to look at genes and characteristics, etc., you'll end up with no embryos to transfer. There'll be nothing suitable. So you'd have to choose what would be most suitable from the batch of embryos that you pick. The use of stem cells to make eggs would be the main thing. Men produce millions of sperm in every ejaculate. We don't need to worry. It's always a question I ask everyone: how many sperm do you think men produce in one ejaculate? And the answer is, hundreds of million. How many eggs does a woman produce in her entire life? She ovulates about 400 to 500, and some of those will not be viable. So the biggest change will be making eggs from stem cells, but that is a really unknown. That is our piece of string. We do not know when that would be possible. There are exciting work carried out in another species, but the humans are very different. I absolutely agree, Nessa, about this technology is not going to be something that everyone is going to be able to access. It's, as I said, people can't even access IVF when they're infertile. So what we are going to end up with, it's a huge class divide, which is already happening. There are already women who could, I think, carry their own children, but are having a surrogate to carry their child, because why should I carry it? I can pay someone $100,000 to carry my child. So I think this technology is going to make even a bigger class divide. We all want to do the best for our children. We all want to give them the best opportunities we can. If you can afford a private school, if you can afford music teachers, dance teachers, etc. So if you can afford to change their genome, then those people will do it. So I think we often talk about the top 1%, and I think they will be taken into a different league. In Jamie's book, he talks about the fact that in the next, children that come back in 100 years, I can't remember the numbers that you used, Jamie, but certainly up until now, we have not manipulated our genome. And I think, Nessa, we do have an absolute difference between what we are doing now with PGT. We're not messing around with the genome. We're taking out some cells of an embryo, and we are testing those cells and putting that embryo back if we feel it's okay. I have real hesitancy about the use of genome editing. I do not know, and we have discussed this a lot in our community, how would we test to see whether those embryos are safe for future generations? And if anyone can tell me how we can do that, I'd really like to hear from them, because we have discussed this so many times. And so when Jamie talked about the mitochondrial transfer being proven, you know, it's not proven. I don't know how we do it. We can't test for generations. So I think we have a lot to think about. I wonder what Darwin will think of us narrowing our genetic pool. We will certainly be narrowing our genetic pool. And I think my biggest concern is really been the safety of this and the big divide it will be between those that have finances and those that don't.
So I'm sure.
David,
all right.
Can I jump in a minute?
Yes, Nessa.
For something there. We will never be able to prove this technology is safe.
Exactly.
Completely safe. But we are never able to prove any technology is completely safe. 40 years after Louise Brown, there are still debates about whether or not in vitro fertilization technologies lead to a higher level of fetal and childhood abnormalities and adverse adult outcomes. We're still arguing about that. There is no technology we can ever create that is safe. In the same way as in drug discovery, we can never create a drug that we can guarantee is safe. All we can do is say that we believe the benefits outweigh the risks and that there is therefore a justifiable approach. So surely we're always in that position with every clinical intervention.
Let's move on, if I can, to taking some of the questions from the audience as well, because I think they add extra flavor. I'd like to come to a question about an alternative approach on the technology. It was raised by Mike Diverd. I'm not sure, sorry, Mike, but your surname, how to pronounce your surname. You're suggesting we should be looking at epigenetic changes more than genetic changes. I'll read the question. Dr. David Sinclair's lab in Harvard has made some experiments indicating that epigenetic programming may be more effective than gene therapy. And as we've heard, CRISPR isn't just about changing the genome. Variants of it can be applied to changing which genes are enabled or disabled. Individual genes may implement multiple separate processes, so influencing genes may be more productive. And if you think it may overtake the use of gene editing?
Can I take that one? Because epigenetics is one of my things.
Yep.
It's extraordinarily difficult to manipulate the epigenome in a way that is consistent and can be maintained. The epigenome is a whole sequence of modifications to DNA, many of which, but not all of which, are removed during the reproductive cycle. We have even less idea about what epigenetic changes do compared with genetic change. It would be much harder to target. The attraction of it is that you're not changing the genetic code. So it sounds like it's a lovely thing to do and it would solve all our problems. Technically, it's phenomenally difficult, and it's hard to see that being reduced to practice in terms of treating genetic diseases in particular anytime soon.
And I would, yeah, but I would just add to that. We haven't talked about these, I mean, everyone will agree with this, but we haven't talked about systems biology. It's a shorthand for us to say we have these systems. We have genetics, we have epigenetics, we have microbiome, virome, all of the omes. But we all know that the body is an ecosystem that's constantly changing, existing within the broader environmental ecosystem around us. And so it's exactly right that all of these other systems will have some bearing. And the more that we learn, the more that we are going to both recognize the greater complexity, and the more that we'll have the tools and knowledge and technology to deal with that complexity. And so one of the reasons why, I mean, certainly there's a debate about whether genetics is special. And I think it is in a way, because it's the blueprint, the recipe, whatever the language people use. But we're going to have to learn about all of these complex systems, and in making interventions, one of the reasons why I think Joyce's word of caution is appropriate is that there are changes that we may make, and even changes where we know we're saving somebody's life, but we don't know all the second, third, fourth, fifth order implications. But there's a debate about Do we have an omnigenic system where all of our genes are doing so many different things that even if there's one instance where we know a certain mutation is causing some terrible thing, that's not the only activity that gene or that mutation may be thing that it may be doing. And that's, anyway, it's a word for caution. And it's why we need to think about cost-benefit analysis when thinking about all of these interventions, and why when there are single gene mutations that are causing outsized harms, we can be more comfortable making those changes because we know that the alternative is something terrible. But the more, it's connected to Nessa's question earlier, the more we start making changes and broader changes, the more unintended or unknown consequences we're going to see.
So 2V Orbach in the chat has said, well, actually, we can improve our epigenetics in many healthy, safe, and natural ways by improving our lifestyle. Recommends there's a lot to be done there rather than delving into potentially unclear, complicated, unsafe mechanisms of CRISPR.
But I'm trying to respond to that by text, but it's actually really slow. It comes back to the point that Jamie raised of overwhelming genetic consequence. There are certain mutations which inevitably have a devastating consequence for the individual. You cannot prevent somebody developing symptoms of Huntington's disease by a healthy lifestyle. If somebody has symptomatic sickle cell disease, same thing. So I would never say don't eat healthily. Absolutely do the most basic things of not smoking, eat lots of vegetables, go for exercise, control your weight, don't overindulge in drugs and drink. But there are certain genetic mutations which are so overwhelming in their impact that there is nothing we can do about those through improvements in lifestyle.
But Nessa, I think they were talking about the epigenetics. They weren't talking about genetics, they were talking about epigenetics.
Well, they're saying you can improve the epigenetics by lifestyle. The problem is we assume that's true. We don't actually know it. You cannot, for example, demonstrate that if somebody eats more green vegetables, they will get lots of healthy epigenetic changes, because we don't understand most of the time how to read the epigenetic changes. But I'm sure they will happen. They will also happen if I eat crisps. It will be very difficult to differentiate between the healthy and the unhealthy. We can't target epigenetic changes. We are still learning, but we do know about the Barker hypothesis, and we do know what happens to you in utero will seriously affect your health, and your birth rate seriously affects your health, and that is related to epigenetics. Oh yeah, I don't debate that whatsoever. I'm just saying we are seeing claims for epigenetics along the lines of eat more of substance X, or the classic one is always drink red wine because it contains resveratrol, and that will work on your sirtuin histone modifiers, and then you'll be much healthier. And actually, we don't have all the links to demonstrate that that is actually true. But epigenetics plays a huge role.
Let's try and take one or two more questions. I'll just remind the audience, you can vote for the questions by hovering your mouse over the questions and clicking them up. We might have time, if we're quick, to take 3 more questions. The top one is on age reversal. If there are genetic or epigenetic implications for how long and how healthily people live, as seems to be the case. I mean, Nir Barzilai in Albert Einstein College in New York has this research project into super agers, people who live to 95 with remarkably good health, little incidence of cancer, health, heart disease, dementia, or diabetes. And it often seems to be some genetic familial connections there. So if we could find out what's in their genetics, is there a possibility that we will select people in the future for longer lives, or even modify our own genetics for longer lives?
Yeah, thanks, David. Let me take, if you don't mind, a first crack at that one. I have a chapter in Hacking Darwin about this where I go through all of the science, and Nir Barzilai and David Sinclair are both good friends of mine, so we talk about this all the time. So let me take it in stages. First, connecting to the question that was asked about epigenetics. Absolutely, all of the smart lifestyle decisions that Nessa mentioned, that's the first step. Everybody, we're born with a natural range of age possibility, and it's different for everybody, and that's largely based on our genetics. but the decisions that we make over the course of our lives can certainly help us be at the higher range of our possibility. And so we don't need to go through it. Everybody knows it. Just live your life like the blue zone people, and you'll be better off. But nobody coming to a futurist call is going to be happy with that answer. So then there's the next phase. What are the things that we can do that are more aggressive interventions? And most of that comes from our knowledge that we have this built-in, essentially survival mechanism in our biology. And the reason for that is our ancestors are the ones who went through all these near starvation funnels when most of the other members of our species, whether this species or precursor species, died. And so we were the ones who we were like those roundworms that kind of struggled on in the petri dish when the food was being removed. And so those genetics got passed on. It also means that we have these triggers, these survival triggers, that we go basically into screensaver mode when we get environmental triggers, we're moving into less and less proven stuff, but when our bodies sense that times are tough, we have a natural process of going into screensaver mode, conserving energy. And so then there are all these drugs that are in one form or another trying to trick our bodies into doing that. Metformin, rapamycin, NMN, NR. It's all that same category of thing. And David Sinclair, the way he would describe what NMN and NR does, it's just improving our copy, our body's ability to make copies of our genetics and our cells. And so that's one whole category. What NEAR and others are doing with their Welderly programs and centenarian programs is they're sequencing these superagers. And definitely, I think personally that we're going to be able to identify what are genetic patterns that if you have them, give you a better chance of living a long and healthy life. We don't have enough, the database isn't big enough, but we're going to have in short order billions of people whose genotypic and phenotypic information are going to be in these massive databases, and we're going to have these incredible AI tools that will allow us to understand that kind of complexity that's very difficult to understand now. So I think we're going to be able to understand that. And then when we can identify the genetic profiles of superagers, it may be that their genes are instructing their cells to do something, and we can mimic that thing. That's a possibility. There's the work in parabiosis. Don't try this at home, where you cut open the old mouse and the young mouse and tie them together, and they're indicators that sew them together, that the young mouse starts to, in some ways, seem older, and the old mouse starts to seem younger. So there's something that's happening in blood plasma, and there are insights there. So this technology is certainly advancing. I think that there will be real tangible benefits. I think that in 10 years, we'll all be taking some kind of precision health extension pill, and probably it'll have some kind of variant of metformin and rapamycin and NMN and maybe lots of other things. David was on in a conference that I spoke at a couple of weeks ago, and they went through, I mean, there were like 80 different ways. And the basic point is that biology is hackable, and it's hackable in a lot of different ways. And we're going to find those things, but it's not going to be, we're magically going to live forever. We're hopefully going to keep making incremental increases to extend our health span, which is really the name of the game.
So if I could add to that, I'm very excited about the idea of healthy aging. Having a population that ages healthily is a really good thing, because it's the chronic diseases of later age which put such a burden, for example, on healthcare systems. I think we have to be extraordinarily careful as a species if we start talking about what can we do to make sure all humans live longer. And the reason for that is that population growth, which we all recognize as a problem, is not driven by birth rates. It's not because everyone's having more babies. They're not. Birth rates throughout the world have been dropping really dramatically. The reason the human population is growing so fast is we are not dying the way we used to. Now, I'm 56. I have no intention of volunteering to lead the shock groups of those willing to die a bit younger. But as a species, we do have to think, why on hell would we want to increase our species' lifespan when the numbers of us actually are having a devastating effect on the planet anyway? So I think this is a real careful what we wish for, because we will just create an even worse world. And especially if we keep using it as badly as we do and with such inequality throughout the world.
I have a complete and strong rejection of that thought, because those are our parents, those are our grandparents. How wonderful if we can extend healthy lifespan to have the wisdom, the love, the nurturing, all of those things that those generations provide. And I certainly, a lot of the growth of our human population is coming from underdevelopment, lack of education. The growth numbers are not coming from the well-educated, developed world where this science is most easily accessible. It's coming from the poorest parts of the world where having more kids is the best insurance policy that you can have, because everything else you don't trust. So I definitely...
The numbers don't support that, Jamie. As soon as women have access to education and have access to choices, one of the first things they go for is birth control, and the numbers drop. Women throughout the world are desperately trying to have fewer children. We also have to bear in mind that we may say that it's the less developed world which is driving population. It isn't, because it's actually the numbers in the West who are driving population, in the more developed worlds where we're not seeing people die as young. But the other thing we have to remember is it's not just about the numbers of people, it's about the impact that we have on the planet. A child in America has something like a carbon footprint that's something like 40 times that of a child in Bangladesh. So we can't separate these issues. It's not a straightforward one. And yes, we'd all like personally to live longer. We'd all like personally our parents, our grandparents to live longer, but we never want anyone else's to. And we have to start thinking about how we create an equitable society. I'm not saying get rid of old people. I'm practically there myself. Please don't get rid of old people.
Can I bring in a quick comment?
Please do, Joyce. And then we'll get on to artificial wombs in the next question.
Oh, yes, excellent. Just in one question. So one thing I talk about with our fertility education, the fertility rate, which is the average number of children a woman has in the EU, is 1.6. And in some countries, it's down to about 1.2, 1.3. So when I was young, women had 2.4 kids. Now in many countries, they're having 1.3. So we're almost at a one-child policy that's just society's choice. So I think we have to think about that, because that's not good for countries economically. But I think I did Google Jamie's age. I think I'm the oldest. I'm not sure how old David is, but I think we're all of that age. I think we're a bit of a select panel. I think as we get to our 50s, we do start becoming a little bit more concerned about aging. And I think this is something that certainly appears on my mind. I know Jamie exercised a lot. I exercise every single day. I do not want to get old. I'm trying to pretend that I'm 18.
Well, I'm definitely old. I'm 61.
Oh, you win. You win.
I'm biased. I've written this book on the abolition of aging, where I do look at this. But if I was in Birkbeck College now, I would say, let's stop this discussion because we can continue it in the pub. Which is what we usually did. Now, what I'm going to do instead of being in the pub, let's see if I can do this right. I'm going to put a Zoom link into the chat for a few people who want to continue some of this discussion from about 5:45 onwards. I'm expecting most of you will be too busy with other commitments, but at least some in the audience will be able to join that Zoom call, a bit like being in the pub, and we can dive into some of these discussions informally. I'll put that link again later on. I would like, however, in the next 6 or so minutes to try and address 2 more very good questions, which at least have got some votes. I'd like to do the question from Diane Ziliox on artificial wombs. Joyce mentioned artificial wombs. Is there anything germane to genetic engineering that would improve the pregnancy experience for women, artificial wombs or otherwise? I mean, you did talk about the live birth rates and what could be done there, but I'm sure you've got lots more to say on that, Joyce.
So I did do a BBC World Service crowd science program on artificial wombs, and it was such an interesting concept. So what we do have right now is uterus transplants. So especially one group in Sweden have become incredibly successful. They've got, well, one of their talks I went to, they had a 100% success rate with womb transplants. And they did also say that it would be technically possible to do this in a man. So I'm waiting for the first, probably be a trans woman who will want to be the first person to do this in a originally male body. But from a feminist point of view, I did say that in a few interviews, I have said that I think one of the most wonderful things about being a woman is that we carry a child and give birth to a child. I can tell you I've had 2 pregnancies, the most difficult months of my life, but I do think that it's a wondrous thing. And in Brave New World, Aldous Huxley took that away and made the pure gender equality. That's the only way we'll have gender equality, when we do that. So I think there's a lot to discuss about the ethics of this.
Anybody else want to comment on that question? I think there's a lot to be done too, and in due course, ectogenesis, when it's possible, it's unclear. 50 years time, a bit less perhaps, I think will transform. I often, in the pubs after London Futurists meetings, do have vigorous discussions between groups of people. Some say that artificial wombs is the most horrible idea ever, and others, usually women, say it isn't too bad, you know, they might be interested in it. So, but I don't want to stereotype the divisions too sharply. The other question I would like to come to is to return to the field of international politics, because that was touched on in the beginning. I feel it's an incredibly important topic. Neil asks, given the present regulatory frameworks within much of the G20, are we at risk of brain drain to locations where genetic engineering regulation is minimal? So not just health tourism, but maybe people will go to countries where they're able to do more research and where they might be able to have children with characteristics not allowed in, for example, some of the G20 countries.
Yeah, so there's 2 points that are connected. One is brain drain, and the other is medical tourism. Certainly on brain drain, we're already seeing that. When in the United States, we had our restrictions in the George W. Bush administration on stem cell research. A lot of our top researchers went to the UK, they went to Singapore, some of them are still there. Certainly there will be medical tourism. And so in terms of scientific transfer of people who are doing serious, credible research, going to places where they can do that research, that's actually something that we want. But we also must have, for something as sensitive as this, we need to have some kind of global governance and regulatory framework. That doesn't mean controlling everything, but we have to, as a collective humanity, at very least try to define red lines beyond which everybody shouldn't go. Because we don't want what we now see with stem cell tourism, where people are going to unregulated parts of the world. They're being harmed by these really horrible procedures. We're talking about the future of life. And that's why there's a race between this technology that's advancing extremely rapidly and our ability to build a moral, ethical, normative, and ultimately regulatory framework on both national and a global level that can establish some kind of guardrails around where we're going. And that's why processes, conversations like this are so important. That's why the work that we're doing with the WHO is so important, but it's really a drop in the bucket compared to what's needed. And so we really need to take this kind of conversation onto a much bigger scale.
And you're doing that in One Shared World, is that correct, Jamie?
Well, One Shared World is about, it's broader than just these issues of genome editing or genetics or even biology. We're with One Shared World, we're looking at the superstructure of the world and say, how do we, for all of these problems where there's a collective action problem, what does the superstructure look like where we can balance our collective human global needs as well as the needs of our countries, and because and other institutions, and because that balance is off, because our countries have such strong representation to almost to the exclusion. That's why the United Nations can't function as it was imagined. We have to rebalance that so that our collective voice and our collective democratically derived needs as humans have a seat at the table. And right now that doesn't exist.
If I could just add to that, David, as well, I think one thing we have to remember is the basic technology behind genome editing is actually extraordinarily straightforward. This is one of the things that's most worrying about gene editing, is that the technology underlying it is getting better all the time. It doesn't require complex reagents particularly, it doesn't require complex experimental setups. So we have a technology with extraordinary potential that it's actually very difficult to restrict access to. In terms of restricting access in humans, most of the regulation of that, I think, will have to come through the reproductive technologies. There are many, many countries throughout the world that could quite easily edit a human genome, edit a human embryo tomorrow, if they could get their hands on it. What they would really struggle to do would be to maintain that embryo and then to implant it back into a human. They don't have access to the reproductive technologies. Where this will become even more of an issue is in non-human uses of gene editing. It's very difficult, for example, to entirely find ways of preventing using this, say, for pathogenizing bacteria or viruses. So I think we have to bear in mind that actually the thing that will act as more as a barrier here, and an appropriate barrier, is the regulation of reproductive technologies, not the regulation of gene editing itself. It's almost impossible to prevent anyone who wants to access it using gene editing.
Indeed, synthetic biology has come along in leaps and bounds, so the things which previously somebody might get a PhD for achieving can now be done almost in just a couple of weeks at home. And there's this competition, IGEM, International Genetically Edited Machine Competition, which has fairly young people doing remarkable things. And we can see that it's nice that they make parts of one species glow in the colors of other species, but the power is there and the dangers are surely spreading. Joyce, do you have comments on the race to the bottom? Are we in danger of missing out on a race to the bottom, or do you have hope that good sense can prevail internationally as well as nationally?
I've never been totally convinced that we can control this internationally. I hope we can. I think, Jamie, in your book, I think you talked about a boat in the middle of the ocean. Did you? I think you did, and that, you know, that has come up before. There are pockets of this planet where you can do whatever you want. Many years ago, I was at a lecture by a very famous UK geneticist, John Byrne, and he said, it's not as if there's mad scientists in a lab somewhere doing weird things. There really is. There are many mad scientists in labs doing weird things. So I just wrote a note there about being cloned. Oh, did I write cloned? No, I wrote cloned. Many years ago, there were one of the leaders of this was Antonori, and also Panos, that said that they'd cloned humans. And I know I did meet people that had worked in their labs and said they had been working on it. So that's why I've just put the question out. So I don't know. We never heard anything else. Antonori once said that he had a few women who were pregnant, but that was about 20 years ago. So I don't know. Did he do it and just not tell anyone? I don't know. So I think globally it's going to be very hard for people not to do what they think they want to do. And this is big money. If a leader of a country comes to you and says, right, I want 500 embryos made of me with my sperm or my eggs, then you know, where do we go from there if they haven't already done that? But
of course, the great thing is they will not be identical.
No, they won't. No, for sure. We haven't talked about cloning.
I don't think cloning's the real issue, the real threat, is it? I mean, some people feel very squeamish about it, but there are many worse things that the technology can do and many better things the technology could do.
Yeah, I mean, no one's going to be, even if you clone somebody. I mean, as you've said, we all know identical twins do have genetic similarities, but they also have differences. So, and if we cloned Trump now and put him in someone's womb, God forbid, it would be a very different person.
So because we started a bit late, I'm going to give another 1 minute each to each of the panelists for final comments. I will put in the chat again a copy to that Zoom link. If people click on that in due course, they should be able to get in without any registration. There's no payment. It's simply an informal chance to follow through in small groups some of the discussions which we couldn't finish off in here. I do want to thank all the panelists for touching on a vast range of important topics, but maybe there are some things that haven't quite been emphasized enough, and this is their chance to do it. Let's go around in reverse order, as it were. So, Joyce, what are your concluding remarks? And then Nessa, and then Jamie.
I always feel I'm the other side of the fence in these discussions. Sometimes I'm criticized for not being a technology optimist. Sometimes I'm criticized for being someone who's trying to halt technology. I don't want to halt technology. I just want us to tread cautiously, and I want us to think about what we're doing, because I do think we're at a really important revolutionary point in genetics.
Good. A really important revolutionary point. We need a lot of thinking. Nessa.
Yeah. There is no such thing as good technology or bad technology. There's just technology and what humans do with it. And unfortunately, we do have a tendency to be a really stupid, smart species. So everybody who's on this call, please read as many reliable sources as you can about this technology. Get involved. It's important, and it's happening really quickly, and it's not going away. And please don't just think about the implications for individual humans. Think about all the other applications for which we can use gene editing as well, and get involved in the debate, because this is just a game changer, the like of which we've never seen in biology before.
Is there anything else you would recommend people read, Nessa, apart from your own books and Jamie's books? And I've read two of your books, Nessa, and enjoyed them both very much and found them very accessible. But is there anything else? You said we are a really stupid, smart species, but perhaps if we read more widely, we can lose some of our stupidity.
Do you know what? I'd actually recommend that people start reading not just the science books, but also books on history and politics and economics. My big push at the moment is I believe everybody should read The Price of Inequality by Joseph Stiglitz to start developing their ideas about what kind of world we actually want to be in.
Joseph Stiglitz, the Nobel Prize economist.
Yeah, it's a fabulous book, and it's never been more pertinent, because we have to decide, are we going back to business as usual, or are we going to build a better world when we come out of this COVID outbreak?
Was that The Price of Inequality?
It's called The Price of Inequality. It's
Thank
a fantastic book.
Jamie, final thoughts from you.
So first, thank you to everybody. I agree with so much of what's been said. Joyce, we're all on the same side of the fence. We recognize the power of this technology, and it needs to be used wisely. And I think that anybody who's saying, let's just race forward, all systems go, is dangerous. I also think the people who say we can never use this under any circumstances, that's also dangerous. The answer is in the middle, and we need to have that balance. We need to have the structures that can facilitate that process, which the UK, as I said in the beginning, is an exemplar of. But I think it's another really critically important point is that we all have a role in this process. Nessa said we all need to be educated. It's not just enough to educate ourselves. If we want to have an inclusive global conversation that leads to norms and leads to standards and ultimately regulations and laws, we all need to feel that we are hubs in that conversation. So everybody is the center of something, whether it's the two people or one person quarantined in your apartment, whether it's your family, your colleagues, your faith community, your country, whatever it is, everybody needs to be empowered to be bringing that conversation to a broader community and bringing a broader community into that conversation. That's how we can move forward together, because I completely agree with Nessa. Technology is agnostic. It doesn't come with a built-in value system. It's up to us to infuse our best values into all of the decisions that we make.
And that's very much the spirit of London Futurists too. So I'm glad we ended on that good note. The things that I'll be tabling for discussion in the Zoom room shortly are 3 questions. 1, did you change your mind as a result of anything you heard today? Because if we're not changing our minds, sometimes it's quite worrying. So did you change your mind? Or if not, what would have changed your mind? Secondly, what did you hear that most made you want to say, yes, that's really important. I'm glad it's being said. We should say it more. And third, what did you hear that most made you want to say, no, I disagree. That's ignorant, dangerous, foolish, misguided thought. And we'll have that discussion elsewhere, but I'll drop that link in one more time. You should be able to get in. Crowdcast says I've already sent that message. Yes, so I have. It's not letting me send it at all. It thinks I'm spamming. Well, this might be the last time I use Crowdcast. I'm not sure.
Shall I send it? I can send it.
Yes, maybe you can. Maybe I've got to say, okay, that's what it is. Thank you, Joyce. And thank you very much for the panelists. Thank you to everybody who asked questions. We only took a few questions, but I'm looking forward to talking with you all again. Thank you, Nessa. Thank you, Joyce. Thank you, Jamie. When I exit this, we'll all disappear. We won't be able to talk to each other anymore. So let's just wave now. Thank you.
Thanks, everybody.
Thank you. It's great.
Thank you.
Bye-bye.
Bye.
Bye.